once users have created groups as shown in the Creating Sample Groupings for Analysis tutorial. The groups are displayed in the Groupings panel. The user can then select groups to perform a differential gene expression analysis on by ticking them. To perform the differential expression analysis, select the statistical test from the drop-down menu located above the list of groups in the Groupings panel. Right-click to perform differential gene expression. The progress of the analysis is shown in the bottom left corner. The results of the differential gene expression analysis are displayed in columns in the Genes panel as rank, genes, number of cells expressing, the group, being 1 and 2, the p-value, the p-adjust, and the fold change. To adjust the p-value, right-click on the Gene panel and select Adjust p-value. Select the desired p-value adjustment method in the drop-down menu and click Set. Once completed, the p-adjusted value will be displayed in the Genes panel. Genes can be sorted in the Genes panel using the right-click Sort By function. Differentially expressed genes can also be sorted by Group 1 or 2. By selecting all genes and then clicking either Group 1 or Group 2. It is very important to ensure that there is no crossover in samples between the two groups being compared. Otherwise, the differential gene expression analysis cannot be performed.